my name is Renee Bishop. I'm from BCM District in the Eastern Cape, and I'm a subject advisor for life sciences. Welcome to the East Cape Department of Education broadcast studios. Today's lesson is part 2 of the scientific investigation, which is a very important topic in both paper 1 and paper 2, as well as being important from grade 10 to grade 12. In part 1, we covered the first 5 common questions that we are asked in the paper, and in today's lesson we will cover questions 6 to 10 of the scientific investigation. So let's get on with today's lesson. Okay, our first question in today's lesson is reliability. Reliability is one of those questions that is found in just about every single exam paper. And it's also one that learners are getting wrong in every single exam paper. They are confusing reliability with validity. And we shouldn't be doing this because it's so easy to remember. Right, let's look at what is reliability. Now, the first thing to remember is reliability starts with an R, okay? And that is going to give us the clue as to what factors cause an investigation to be reliable. Now, reliability has to do with how many times I do something. Because the more times I do something in the same way and I get the same answer, the more reliable something is. It's like when you ask your friend to help you. You know your friend is going to say yes every time because they are reliable. So getting the same answer many times gives us reliability. So let's look at the factors in an investigation that make it reliable. Right, the first one is repeat the investigation. Notice repeat starts with an R. Okay, we're emphasizing the R's here for reliability. So repeat starts with an R. Repeating the investigation means that I'm doing the investigation again. I'm repeating the whole one. Maybe I'm doing it in another place. I'm doing it with other people, but I'm redoing the investigation. I can also increase the length of time for that investigation to make it more reliable. This is not always the case because some investigations won't become more reliable if we increase time, while others will. The second thing we can do to increase reliability is use a large sample size. You see, I even found an R for you there. Okay, a large sample size. So using one sample is not reliable because you might get wrong results. But using more samples, 2, 3, 4, all right, samples will make it more reliable. We have to, however, be careful that we don't use too many samples that makes it unmanageable for us. So scientists need to consider how big their sample must be, right? It needs to be sure that it is reliable. The third factor is taking several samples or readings, okay? By taking several samples or readings, all right, you are increasing the number of readings you take and therefore making it more reliable. Usually we take a number of readings and we find an average reading for the investigation. The last thing that makes an investigation reliable is random sampling, right? Choosing your samples randomly, not selecting those that you want, okay? Because we may be biased to certain samples and then we might affect our results because we are choosing specific samples. Right, so let's have a look at our trial paper from 2024 and let's look at how the investigators made this investigation reliable. Okay, so firstly, remember, okay, if we look back, this investigation was about microplastic bioaccumulation on the male fertility in rats. Okay, and we are going to look here at the procedure, these dots, okay? That's where we're looking in this investigation to see how investigators made this reliable. What did they do? Okay, so the first thing that we see in this investigation is that the investigators used 60 male rats, which they divided equally into 3 groups. In other words, each group had 20 rats. Now, when we write this answer, we don't write they use 60 rats, which is a large sample size, because it does not tell me how many rats were in each group. I might have put 1 rat in one group, I might have put 19 rats in the other group, and 30 rats in the third group. That means that the one group only had 1 sample in, which would make it unreliable. So I need to state that there were 20 rats in each group. All right? That is a large sample size, and that's what made this investigation reliable. The second factor that made this investigation reliable is that the investigators chose 10 rats in each group, all right, and took samples from 10 rats and found an average for those samples. So they took 10 samples from 10 rats, okay, and found an average. The third thing that made this investigation reliable is they randomly chose the rats for their samples. Okay, so if we read here in the investigation, they randomly chose the 10 rats, which makes it reliable. Now you'll notice that when I'm answering this question, I'm not using a generic answer like large sample size. Take many samples and find an average. Okay, I'm giving my answers according to what is written in this investigation. So I'm being specific for this investigation. And that's what examiners are wanting you to do nowadays. Remember, scientific investigation questions are level D questions. They involve analyzing and synthesizing. So we need to be able to apply, all right, to the investigation that we are given in the question paper. So in this investigation, I'd had 20 rats, which is a large sample size. I took 10 rats and took a sample from each one. And I chose the rats randomly, all right, to sample. Right, now let's look at another way of asking the same question. Often, they might ask you, instead of saying, how did the scientists make this investigation reliable? They might say, how can we make this investigation more reliable? How can it become more reliable than it already is? Then we need to look at what did they do and what did they not do, okay, to see how they can make it more reliable. So let's look at our trial paper once again. In our trial paper, they took random sampling, right? They took many samples and they found an average. They used a large sample size of 20. Although they could use more than 20, if they used more than 20, it would make the investigation more reliable. But the one thing that they did not do is repeat the investigation. So by repeating this investigation, we are going to make this investigation more reliable. Okay. Right, now it's your turn again. Okay, so with your November paper 1 from 2024 that we were using in part 1 and November paper 2 from 2024, right, we're going to look at questions 3.5 in November paper 1 and 3.3 in November paper 2. And in both these papers, you are going to answer the following questions. Why was the investigation considered reliable? And how can I make the investigation more reliable? Right, let's see if you can do those. Right, welcome back. Let's see how you did with those 2 questions. Okay, firstly, looking at paper 1 from November 2024. In paper 1, their reliability was ensured because they used 4 plants in each group, which is a good sample size. Okay. It can be made more reliable by using more than 4 plants in each group. Now notice I did not say use a large sample size because I need to be specific to this investigation. So if they used 4 plants in each group for this investigation, then they must use more than 4 plants if they want to make it more reliable. Also, I can repeat this investigation to make it more reliable. Right, and let's look at paper 2 now. How did you do there? The first question was, how was it reliable? Here again, we used 2,073 children per group. Now notice here, I did not say 4,146 like it says in the question paper. So in your question paper, it said 4,146 divided into 2 groups, okay? So each group had 2,073 children in it. Okay, remember we have to talk about the size of the group, not the size of the overall sample that we are taking. Okay, so we had a large number of children, 2,073 children per group, which is a large sample size. Also, the investigation was conducted over 7 years, which is a long time period. Notice I'm answering it specifically for this investigation. I do not say it was a long time period. I say it was conducted over 7 years. How can I make this investigation more reliable? I can do it by repeating the investigation that was done. When you say repeating the investigation, there is no more specific way that you can say it, all right? You can say to do it again in another country, all right? To do it again with a new sample of children, all right? To do it again with a different age group, all right? But it is just repeating the investigation. All right, hopefully you've got that down pat. Now that we're going to do validity and we're not going to mix the two up. Right, so let's look at the next 2 questions. Validity, right? Validity starts with a V. A V doesn't look anything like an R, does it? Eh? So we shouldn't be mixing these two, okay? Validity has to do with the variables, okay? The variables in the experiment have to be kept the same, okay? So as soon as they ask us about validity, immediately I must think about variables. Reliability, I think about numbers, okay? Validity, I think about variables, okay? So validity is based on what scientists did in the investigation, okay? What did they keep the same? Now, each investigation is different, okay? So we can't learn standard answers for validity, right? We need to read the investigation, understand the investigation to be able to answer this question. And once again, we're going to find the answers in the procedure, the dots on your page, okay? Right, so I'm just going to give you some clues before we start on the type of things that might affect the results of your experiment, okay? These are factors that should be kept constant or should be controlled. Right, so things like age, sex, the species of plant or animal, the environmental conditions such as light, sunlight, the cage size that the animals are kept in, right? The amount of liquid that we are testing, the equipment that we are using, and the size. Maybe it's the size of the cage, maybe it's the size of the animal or the plant that we are using. So these words that I'm giving you are just there to help you think. They are not answers that we can use. Right, so firstly, we're going to look at our investigation, all right? And we are going to look at what variables were controlled. In other words, how was validity ensured in this investigation? How did we ensure validity, right? And if I want to explain that, I will say, what variables were kept constant. So have a look at your dots, okay? And let's look at what did they keep constant. Firstly, all the rats in this investigation were from the same species. They were all albino Wistar species. Secondly, they were all healthy rats, okay? Also, they were all fed the same diet. The investigation was carried out for the same period of time, which was 90 days. If we look there, they did the investigation over 90 days. So all groups were tested for 90 days. And also, we tested each group every 30 days. So the time period for testing was the same. So these factors were controlled in our investigation. Now, as I mentioned in part 1 of the series, all right, I have not used male or gender as a factor that was controlled. Okay? Although all the rats in this investigation were male, I cannot do this investigation on females because females, we don't measure testosterone level to determine their fertility. All right, so we can only use males for this investigation. Therefore, males is not a controlled factor, okay, in this investigation. Also, I haven't mentioned that the number of rats in each group was the same because very often in an investigation, all right, they don't consider the number in each investigation being the same as making the experiment more valid. Okay, so in some papers it has been credited, whereas in other papers it hasn't been credited. So try to look for other factors that are kept the same in your investigation. Right, now the other way of asking this question is how can we improve the validity of this investigation? How can it be improved? In other words, what variables were not kept the same but can affect the results of your investigation? Now, when you are trying to think of variables that need to be kept constant, think about which variables will affect your results. Now, in the case of the investigation that we're looking at the moment, which is your trial paper 1 from last year with the rats and the microplastic bioaccumulation, our results were testosterone level. That is what we were measuring. So in other words, we need to consider what variables will affect the testosterone level in rats. Okay. Now remember I gave you a couple of cue words, okay? These were some of the cue words that I gave you to think about, all right? So when I'm trying to select which variables must still be kept constant, I can use these cue words to help me to work out which should be kept constant. So let's look at my cue words. My first cue word was age. Was the age of the rats kept constant in this investigation? And if we look here at the procedure, we'll see it wasn't kept constant anywhere in this procedure. Okay, so we can still improve this investigation, make it more valid by keeping the age of the rats the same, because age affects testosterone level. If I took a rat that was 1 year old compared to a rat that was maybe 2 years old, okay, the age affects testosterone level because testosterone increases when males become reproductively active. Right, the second cue word that I gave you was the sex or the gender of the animal. Now, in this investigation, they were all male rats, okay? So it's no need to control gender because we are looking at male fertility, right? So this one cannot be considered as a factor that we need to control. The third one was species. Now, the species of rat is already controlled in this investigation. So it's not a new factor that we can still control. The next one was environmental condition. Environmental conditions are things like temperature, sunlight, the size of the cage. Now we need to think about what factors will affect testosterone level. Will sunlight affect testosterone level? I don't think so. Okay, all right, but maybe temperature will affect testosterone level. Okay, perhaps the stress of the cage will affect the testosterone level. So there are environmental factors that were not controlled that could affect the testosterone level. Okay? Right, then the next one was the amount of water, the amount of the liquid being given. In this investigation, they were given water that contained different amounts of microplastics, but they didn't control how much water they gave each of the rats every day. Okay, and that would affect how much microplastic the rat gets in and therefore affect the testosterone level. So they should have controlled the amount of water that they gave the rat. Right, equipment or method of testing. How did they measure the testosterone level and how did this affect the rats? Right, it might have affected the way that testosterone was extracted from the rat, may have affected how much testosterone was extracted. So we need to keep the method of testing, all right, the same. Then the size. Does the size of the rat matter for testosterone level? I don't think so. Right, whether the rat is big or small will not affect the amount of testosterone. The age will, because we need to look at reproductive age, but the size of the rat won't matter. Then the person doing the testing, this might have an effect, sometimes it does, but if the procedure is a standard procedure, then the person doing the testing, it does not affect the results. Okay, so in this question, how can validity be improved? Right, we have, firstly, we could have kept the age the same. We could have kept environmental factors such as temperature and cage size the same. We could have kept the amount of water the same. And we could have kept the equipment and the method of testing the same. Right, so it's your turn again, okay? All right, we're going to be looking here at November paper 1 and 2 again. And in both papers, you are going to answer 2 questions. Firstly, how did scientists ensure validity in this question? And 2, how can this investigation be made more valid? Right, let's answer those quickly. All right, welcome back. Okay, let's see if you got this right. Right, our first question in paper 1 was, why was this investigation valid? Now, if we look at paper 1 and we look at the procedure over here, okay, we will see that it was valid because firstly, the plants in the sample were all of the same species. Also, the plants were all kept in darkness. And thirdly, they were all kept in darkness for 72 hours. Okay, the same duration, the same length of time. We can make this investigation more valid, okay, if the plants were all the same height when we started, because this might affect how close they are getting to the light. Okay. Also, the environmental temperature and conditions could be kept the same. Right? Things like temperature, humidity might affect the growth of the plant as well, and therefore affect how the plant responds. Right, also, if plants were given the same amount of water, right, and treated the same way, for instance, had the same soil conditions as well, right, this might also affect the growth of the plants and affect our results. Right, and then paper 2, let's say how you did in paper 2. Right, in paper 2, we once again look at the first question. How was this experiment made valid? And in paper 2, wow, these scientists really looked at making this paper valid. Right, they kept everything the same. We have a long list of things that were kept the same. Right, let's go through them. Firstly, all the children were healthy. Okay. They were all 3 years old. In other words, they were all the same age. They were all the same race. They were all the same gender. They don't tell us which race and gender, so we cannot say which race and gender in the answer. We just state they were all the same gender. They were all given 1 liter of milk daily. They all had the same additional diet. They used the same supplier of milk. They had the same duration of the investigation. They had similar daily activities, and they had similar living conditions. Wow, that's a long list of things. So in other words, the scientists controlled many factors here to make this experiment valid. Okay, so it's got a high degree of validity in this experiment. Let's look at that second one now. Can this experiment be made more valid? All right. There's very few things left, but perhaps we can look at genetics. The genetics of the parent will also affect the results. Remember, when controlling factors or variables for validity, we need to look at what variables will affect the results. And our results are the height of the children. So we need to consider what variables will affect the children's height. And genetics plays an important role in height as well, right? Also, the same height at the start of the investigation, before they started giving them the specific type of milk. Surely the children should have been the same height. Okay, so those are 2 factors that might improve the validity of this investigation. Right, and today's last question is question 10, which is the control and baseline. What is a control and what is a baseline? Okay, these are things that are, or terms that are not found in our textbooks, all right, but they are often asked in exam papers, and they are very important parts of an investigation. Firstly, a control is a group in the experiment that does not receive the independent variable. So if we have 2 or 3 groups in our investigation, there should always be one group that does not contain the independent variable. In the case of our experiment, we will, in the trial paper that we spoke about, the microplastic bioaccumulation in rats, our microplastic bioaccumulation was our independent variable. So if I want to make a control, I need to have a group that does not receive microplastics, and there is no microplastic bioaccumulation in them. Right? A baseline, on the other hand, is a measurement taken before the treatment of the independent variable. Okay, so in other words, a baseline, if I am investigating the effect of eating on the glucose level in our blood, I will measure the glucose level of the blood before I eat. All right, so before I eat, I take a measurement, and that will be my baseline, so that I can compare as to what happens after I'm eating and how glucose level changes. So both these factors, both a control and a baseline, serve as a point of reference in our investigation, so that we can compare them to the other groups or experiments, okay, and to see if it is the independent variable that is causing the results that we are obtaining. Okay, so let's practice that now. All right, let's look at the control. Here I have an experiment, especially for the grade 10s watching. All right, In grade 10, we did the photosynthesis experiment. I'm sure the grade 12s can remember this one, where we took a leaf, all right, and we covered it with a piece of tin foil, all right, so that some of the leaf was exposed to light and some of the leaf was not exposed to light. The aim of this investigation was to see if light is needed for photosynthesis. In this investigation, my independent variable is the light. Okay, so if I look at this leaf, there are two parts. There's this green part exposed to the light and the silver part that's covered with aluminium foil that has no light. The green part, the part exposed to light, is the experimental part. This part that's covered, that has no light, in other words, no independent variable, okay, which is light. This part that is covered, that part is my control, okay? And remember that when we tested this experiment in our investigations, we found that starch is produced in this part, in the green part, all right, because the iodine turned blue-black and told us that starch was being produced. In this area, okay, that was not covered, all right, the iodine remained yellow-brown, telling us that no starch was produced. So what does this tell me? It tells me that light is needed in order for photosynthesis to take place. And how do I know that? Because starch was produced. Starch was produced, telling me that photosynthesis takes place in this top part of the leaf. The control, however, that had no light, produced no starch. Therefore, no photosynthesis took place. And therefore, it proves my point that light is required for photosynthesis. So the control is proving that point to me, that it is the light for the independent variable that is causing the results or the production of starch in this case. So let's put that into words, okay? You can learn this definition, all right, and use this definition when you are explaining what a control is used for. So why is a control needed? A control is to compare with the other experiments or groups to show that it is the independent variable that caused the effect or the result and not any other variable. Okay, the baseline, okay, is very similar to control, but it is not a different experiment that does not have the independent variable. Remember, a baseline just is a reading you take before you administer the independent variable. So once again, it's to compare with the other readings to show that it is the independent variable that is causing the effect or the results. Okay, so let's look at our trial paper from 2024 and let's look at where the control is and what is the purpose of the control. Now, if I look at this experiment, I see that there are three groups, 1, 2, and 3. Okay? My first question is, which group is the control? Now remember, the control does not have the independent variable. So I need to identify the independent variable first. I go look at my aim. And in my aim, it says microplastic bioaccumulation on male fertility in rats. Microplastic bioaccumulation is my independent variable. So, which group has no microplastic bioaccumulation? It is group 1, because we see there that there is no microplastic added to their water, therefore no microplastic bioaccumulation over the period of 90 days. Group 1 is my control because it has no microplastic bioaccumulation. Right, then why was it included in the investigation? Why did I make this group with no microplastic in it? Okay, firstly, it was to compare with the other groups to show that it was the microplastic bioaccumulation, in other words, my independent variable. Notice that I've replaced the word independent variable with microplastic bioaccumulation. I've written the independent variable for this investigation. So it's to show that it is the microplastic bioaccumulation that caused the decrease in fertility. This is the result, okay? All right, in my generic one, it said that caused the result. I need to replace that word result with what was the result, the decrease in fertility, and not the water, not the other variable. In this case, it is the water. So let's practice that quickly. All right, explain why Group C was included in the investigation in your November paper 1. Okay, let's check you. Right, explain why Group C was included. Because it is a control. It's there to compare with the other groups, to show that auxin causes the tropism and not the agar. Right, notice how I've replaced the words with the words used in the investigation. Let's practice a baseline now. Okay, answer the question on why is the expected average height of children included in the investigation for November paper 2. And welcome back. Let's see what you got. It's included because it's a baseline. It's there to compare with the other readings to show that it is the dependent variable, in other words, the type of milk that caused the growth in height. Okay, all right. And that brings us to the end of today's lesson. I hope that you are now able to answer the 10 questions, the 10 most common questions that are found in the scientific investigation, right? And that you're no longer making the common mistakes and losing those important marks. Okay, remember to still practice your graphs and tables and calculations, as these are also important parts of your scientific investigation. Right, you can leave your questions on the comments. You can leave your questions on the comments. Don't forget to leave the name of your school so we can give you a shout out. Please share, recommend, subscribe to our YouTube channel. And thank you so much for watching. If you haven't watched part 1, go back and watch part 1. Until our next lesson, good luck for your exams and see you soon. Bye-bye.